Okay, so just so you see kind of how we're going with this class, just to kind of show you that there is, despite the idea that I'm just randomly throwing crap up here, this is how the class has been put together. So we started off talking about how prokaryotes were put together, and then we moved on to talking about how they function, how they moved towards nutrients when we talked about chemotaxis. Then we moved on and talked about once the chemotaxis was successful and got them to an area where there were nutrients, how do they take those nutrients up? Then we moved on to how they break those macromolecules down, so protein secretion, releasing enzymes into the environment to break down macromolecules so that they could take them up. And then how do they use those nutrients to create energy, so our metabolism for generating precursors. And so that brings us now to, once these organisms have found their way to a nutrient source, have broken it down, have taken up monomers, have created energy, what do they do then? They grow. They increase in their cell numbers. So that leads us to this next section that we're doing, and all recognize that this is not on the exam next week. This is a new set of material where we're talking about growth and division. So I don't want to belabor perhaps one of the most boring points of any microbiology course, and that's talking about how microbes divide, because it is really, really dull. Bacteria, divide by binary fission almost exclusively, and so there are a few things that we need to know about it. But mostly we're going to talk about the consequences of bacterial growth, certainly rather than the arithmetic that goes in with these. But generation time, by definition, simply is the time that it takes a population to double. And you've all heard me talk about the fact that bacteria have this advantage in a high metabolic rate, which translates into a high rate of division or a low generation time. So there are bacteria that in the laboratory can double their population every 13 minutes. But if you look at bacterial growth curves in the laboratory, generated in the laboratory, and we'll do this in the lab class, they're usually shown on a semi-log scale, because if you look at how bacteria grow, and we can sit here and do the math together, one bacterium becomes 2, becomes 4, becomes 8, 16, 32. If you graph that arithmetically, what you see is a really slow increase during the early portions of this growth curve, shown by this green line. And then eventually, once the numbers get larger, doubling just makes things go even faster, so you get this asymptotic line. So instead, they're shown on a semi-log scale, where the bacterial cells are measured on a log scale as opposed to an arithmetic time scale. So you'd have a line such as shown here in the red. So bacteria, when they're growing at their maximal rate, when they're not being inhibited by waste products that they've made, when they're not being limited by resources that they've used up in their environment, are growing in this logarithmic phase, so on a straight line on a semi-log scale. You can use these things to measure generation times. Typical generation times go between 20 minutes. That's about what E. coli can do in the laboratory, depending on exactly the conditions you're growing them in, to some that are days or weeks long. I feel very badly for my friends who do research on the bacterium that causes tuberculosis, Mycobacterium tuberculosis. It takes weeks for the bacteria to form colonies on plates. They have very slow generation times. But for the most part, when you have these, my kind of problem with generation times like this is that they're very artificial. These are laboratory artifacts. And bacteria don't really live in the laboratory as part of their natural history. They live in the real world. And just to illustrate the fact that when you see data on growth curves and generation times, think about E. coli, which in the laboratory has about a 20-minute, give or take, doubling time. But in the real world, in its real situation that it finds itself in, for the most part, in a colon of a warm-blooded vertebrate, it can take about 12 hours for a bacterium to divide in that colon. And you can imagine, because there's so much more competition by so many other bacteria, nutrients, are at a premium because other bacteria are taking them up, and waste products from their own brethren bacteria as well as competing bacteria. So take this. This is part of the reason why I don't like to spend a lot of time on the arithmetic behind growth curves. We will do it in the laboratory a little bit, just because anyone who looks at your transcript later on and realizes that you took microbiology will assume that you know something about growth curves and stuff. This is a typical growth curve that you can create with a bacterium in a laboratory. And again, this is a laboratory artifact. This is not how bacteria typically grow in the environment. Because in the laboratory, we start off by putting just a few bacteria, like when you do inoculations into the broth cultures that you've done for last week's lab. You start off with a very low number of bacteria, at least in terms of concentration. And so there's an initial lag phase where the bacteria are just like, holy cow, I'm in a whole new place. What the hell do I do now? In reality, they're not saying that. In reality, what they're doing is they're adapting to that new environment. So they don't begin to grow for a little while. So there's a lag phase. But then once they get with the program, once they realize where they're at and they synthesize the enzymes necessary, they grow in this logarithmic or exponential phase. And this is a bit, for me, of a problem, because most of us, and I include myself in this, when we do research on bacteria, either genetically or physiology in the laboratory, we use bacteria that are growing in this exponential phase. We do it because here, when bacteria are growing in this exponential phase, they are at their most healthy. All of the cells in that population are rapidly dividing, and so it's a very uniform population. And so from a biochemical point of view, it makes studies much easier, much more, there's much less variability. But in reality, this is probably not a situation that most bacteria in their natural environments find themselves in very often. Probably more often than not, this next phase of growth, which we see in the laboratory, which is called stationary phase, which is where you have a balance between bacteria being born and bacteria dying. This is probably more closely related to what microbes encounter in their environment. Just kind of hanging on. Some cells are dividing, but other cells are dying. And in microbiology, we talk about bacterial cell death as simply the inability to divide any longer. They can still have metabolic activity, but they can't divide any longer. Stationary phase, you have a population of bacteria, some of which are actively dividing and others have lost their ability. That's why we don't usually use this population in studies in the laboratory. But in reality, most of the bacteria that are probably existing in our colons right now or on our skin or in our mouths are probably in a population that's not rapidly increasing or decreasing. But then in the laboratory, if you continue the incubation further, you'll go into the death phase, where now the growth is not balanced. You still have bacteria during the death phase actually dividing, but more of them are losing the ability to die. So more are losing the ability to divide. And so those bacteria are actually dead. And it's interesting that some bacteria are selected here because they're actually cannibalizing the dead remains of other bacteria. So as bacteria die and lice, they provide nutrients for bacteria that may be starving. So the other thing that I like about this growth curve is I think you could think about this in terms of human population on Earth. So substitute human population for bacteria population and try and figure out where we're at on here. I think we're sadly still on this exponential phase. I just don't know how close to stationary phase we've got, but that's a matter of personal conjecture. So, as I said, I don't want to spend a lot of time talking about the arithmetic of growth. I want to talk about the consequences of bacterial growth because it's much more interesting, much more applicable. And so as one of the, or as one of the best examples of a consequence of growth, I want to talk about a process that has really come to be widely understood just in the past decade or two. When I was in graduate school, the term quorum sensing didn't exist. There was a term similar to it called autoinduction, but what I want to describe now what's known as quorum sensing. So this is actually really cool when you think about it, as a biological philosopher, if you will. We think of bacteria, even microbiologists to this day, as autonomous little bugs in a population that are completely ignorant of the other bugs that are in that population. But in reality, most bacteria have the ability to behave as if they are a multicellular organism. And that's what quorum sensing is. Quorum sensing is probably the earliest advent of multicellularity that we know of. So this population of bacteria actually has the ability to detect its population size. It has the ability to sense when it has reached a quorum. So those of you who understand parliamentary procedures will understand that you can't do certain things in a group of people unless you have a sufficient number, a quorum of people for the meeting to be official. So bacteria have evolved, many bacteria have evolved the ability to tell when they have reached a critical population, and then they change their behavior. And I put behavior in quotes because by behavior, for the most part, I'm saying gene expression. So bacteria will change the pattern with which they express genes based on the population. And this makes perfect sense if you think about the behaviors that are being changed as a function of population. So many bacteria, and we've talked about secretion already, secrete extracellular enzymes and other molecules like siderophores, those iron-binding molecules, into the environment. If those bacteria in a pioneer population, when the population is very low, if they began to express large amounts of siderophore or large amounts of enzyme, just pumping that out, using their energy to create those things, it wouldn't do that low number of cells very much good because most of that product would diffuse away. And in the case of siderophores, the chances that those molecules would find their way back to that same bacterium that synthesized them are pretty low. If you are a bacterium secreting an extracellular enzyme and you are by yourself, for the most part, that enzyme is going to diffuse away. If it does find a substrate to break down, like starch, say, or a protein, those amino acids from the protein or sugars from the carbohydrate are probably not going to diffuse back to you. So instead, it makes sense that bacteria have evolved the ability to tell when they're at low numbers and to not bother to express high levels of these extracellular enzymes or siderophores. They do express them, but at low levels. Instead, what they're doing is they're biding their time, they're saving their resources, they're putting their resources into dividing, into growing the population. Because they realize that at some high population, at some quorum, if suddenly all of the members of that population begin to express siderophores or express an enzyme, then they get the benefit of all of the other members of that population producing those enzymes or that siderophore. So they get a kind of a community action. This is also very important in pathogenic bacteria. Many bacteria that have the ability to secrete toxins don't do so when they're at low numbers because the toxin molecules aren't going to be able to be produced by just a few bacteria enough to do the bidding of that bacterial population. So better they lie in wait until they've reached a quorum, and then suddenly that population can and will, as a single organism, begin to express large amounts of that toxin. Because now they can take on the host. That toxin produced by 1 million bacteria can do something that toxin produced by 100 bacteria could never do. So they wait for this quorum to be reached to produce toxin. Now, this was initially described and for the longest time thought to be a very unusual property of a bacterium that lives in the ocean. This man is still alive today, Ken Nielsen, who studies microbiology at the University of Southern California. And he described this process whereby bacteria induced themselves to produce luminescence when the population got to be very high. And for a long time, people kind of dismissed Nielsen's work. And then when they did accept that he was doing it correctly, they thought, oh, this whole process is really just restricted to this one weird bioluminescent bacterium that lives in the ocean. What we know now, because Nielsen was very good at persisting, is that the process that he was describing for this marine bacterium, which I'm going to describe in just a little bit, is actually very widespread among gram-negative bacteria. So I want to go through kind of a schematized general mechanism for how gram-negative bacteria can use quorum sensing to detect population changes and then affect their behavior. So in gram-negative bacteria, and this doesn't mean all gram-negative bacteria, but it has been found in a large number of gram-negative. They have the ability to produce what are known as acyl homoserine lactones, or AHLs. These used to be referred to before people knew the chemical nature of them as autoinducers. So you still see the term autoinducer many times meaning a homoserine lactone. This is the chemical nature of these homoserine lactones. They tend to be species specific, which is really cool because 1 gram-negative bacterium can produce a homoserine lactone that has specific substituents on it that make it fit lock and key with only members of that species. So it has the ability to talk just to other members of that particular species, but other species ignore it or are blind to it. But it has a homoserine lactone portion of it, and then attached to that is, in essence, a fatty acid chain. This acyl chain is a fatty acid chain. And this is really important because this acylation, this fatty acid put on there, allows these homoserine lactones produced in the cytoplasm of the cell to diffuse across both plasma membrane and outer membrane. There's no energy required for the export of homoserine lactones. And the other really important point is there is no energy required for them to come back in. So homoserine lactones will freely diffuse out and freely diffuse in. And so there's no barrier to them. And so the concentration inside of the cell is basically equal to that outside of the cell. Now, these homoserine lactones are produced by a synthase, homoserine lactone synthase, which is usually referred to as LUX-I, L-U-X capital I. That was the first gene ever discovered that synthesizes these homoserine lactones. So now we generally refer to these homoserine lactone synthases as members of the LUX-I family of proteins. But this homoserine lactone synthase is produced at low levels constitutively in bacterial populations at low population densities. And they're producing homoserine lactones, which are diffusing out of the cell and diffusing back into the cell. When populations are low, the homoserine lactone levels are low. So think of this homoserine lactone level as a surrogate for population level. As this bacterium now divides into 2, and then into 4, 8, 16, eventually all of those bacteria, all producing small amounts of these homoserine lactones in that environment, create a condition where now the homoserine lactones are diffusing into the cell very rapidly. So concentrations of homoserine lactone actually build up inside of the cell. And many of those individual molecules, in fact, most of those individual molecules were actually synthesized by other bacteria in the population, not being synthesized by the same bacterium. Now, the way that the behavior changes, once the bacterium reaches this quorum, once the bacterium reaches a level of homoserine lactone that says, okay, there's 1 million of us, there's 10 million of us, let's take on this giant carbohydrate that we're stuck against, or let's take on this host that we're pathogens for. The homoserine lactone binds to another member of the LUX family of proteins called LUX-R. So LUX-R, again, was the first gene and the first protein characterized of this family. So we still tend to refer to members of this regulator protein as LUX-R, R stands for regulator. This is a transcriptional regulator protein. So this very simple cartoon shows LuxR in two different forms. Circular form is inactive. At this point, when it exists like this, it can't do anything in terms of transcription, affecting the transcription in that bacterium. But when enough homoserine lactone has diffused in, the pool of LuxR shifts from this circular form to this square form. So it shifts from what's referred to as apo-LuxR, meaning it doesn't have homoserine lactone bound, to the form that has the homoserine lactone bound. And you can see there's a protein conformation change indicated by that change from circle to square. And that in that form bound by homoserine lactone, it has the ability to bind to DNA to upregulate certain genes. So it wouldn't just be binding, as it shows in this cartoon, to one spot. It would be binding to multiple promoters throughout the chromosome and turning those genes on. One of the important things to keep in mind about this pathway is that one of the genes that's going to be turned on by this activated LuxR is the LuxI protein, the homoserine lactone synthase. What happens is this is how the bacterial population really gets the message in a hurry that quorum has been reached. So as this LuxR pool now shifts towards the square form, the bound form, and starts to turn on the transcription of target genes of quorum sensing, one of the first genes that gets upregulated is the synthase itself, and so now more homoserine lactone is being produced. And this is happening simultaneously in the entire population of 1 million or 10 million bacteria. So now there's a sudden really big production of homoserine lactone. That ensures that everybody in that population gets the message almost simultaneously. So now all of these other genes that are being now expressed as messenger RNA and then messenger proteins are occurring almost simultaneously throughout the population. So the population is behaving as if it is a multicellular organism that just got a message from a hormone. In effect, you can think of this as a pheromone. It's been referred to as a pheromone. And now these quorum-dependent proteins are going to help that bacterial population survive that increased concentration. These might be toxins, they might be enzymes. For the most part, this is not a hard and fast rule, but for the most part, these quorum-dependent proteins tend to be proteins that are secreted, because most of the community benefit that a bacterium is going to get from simultaneously having the entire population produce this protein is if those proteins are helping to break down something in the environment, a food source, or if they're a toxin. So more often than not, these quorum-dependent proteins are secreted. Not always, and I'm going to show you the best example for this actually involves bioluminescence. And in that case, these proteins are not secreted. They're used to generate light within these bacteria. So this has become a very well-studied process that's been found in hundreds of different species of bacteria. Not all, but hundreds, perhaps thousands. So very well known, but it's found only among the gram-negative bacteria. So far as I know, there has never been a homoserine lactone synthase found in gram-positive bacteria. So it might seem that gram-positive bacteria like Staphylococcus and Streptococcus don't accomplish quorum sensing. And it's really not true. I put in this slide at the last minute when I was updating it, simply as another way to look at the quorum sensing in gram-negatives. And mostly rather so you don't think of the synthesis of this homoserine lactone as being very simple. It's actually highly complex, all done by this LuxI protein. You don't need to know the individual steps of this or the intermediates, but just showing that these homoserine lactones are put together by multiple steps before they diffuse out. This cartoon is also kind of nice because it shows the LuxR protein existing in the apo form, shown here by this red and tan. And then when it binds to the homoserine lactone, it forms a dimer. This is actually how it becomes activated. The protein conformation changes, but it also forms a dimer. And that dimer, and only in that dimer form, is it capable of binding to DNA regions of the regulon, those genes that need to be turned on, or in some cases turned off, in response to quorum. So this is just perhaps a better cartoon for how homoserine lactones function in quorum sensing in gram-negative bacteria. But as I was starting to go on about, gram-positive bacteria also face the same dilemma in terms of population dynamics. They have to be able to put out toxins or put out enzymes when they reach a critical threshold as well. So gram-positive organisms have actually evolved quorum sensing entirely independently of gram-negatives. I can't tell you how long ago gram-positives and gram-negatives split phylogenetically, but it was a hell of a long time ago. But what I love about the fact that gram-positives have a completely different system is that it shows how critical multicellular behavior is, even to bacteria. It has evolved at least 2 independent times. So the homoserine lactone system that we just described in gram-negatives is very widespread among gram-negatives. The system that I want to describe now is completely unrelated, uses completely different genetic machinery, but it accomplishes the exact same thing, and it's found only among the gram-positives. And it's quite widespread among the gram-positives. So these are ancient pathways that have evolved differently but do the exact same thing. So gram-positive species like staph and strep and bacillus use a system where what's being secreted into the environment is not a homoserine lactone, and it's not even something that can freely diffuse across the membrane. It actually requires energy. It's a peptide. So here in this not very good cartoon is a schematized gram-positive bacterium. And here you have the gene labeled here as PP for peptide pheromone. This gene will be transcribed into messenger RNA, translated into a protein in the cytoplasm, and then that protein will be exported and simultaneously chopped up into little peptides. So what's not being shown here is a secretion pathway that simultaneously secretes the protein, cuts it up into little peptides, these pheromones, which now function exactly the same way that homoserine lactones do. They are a surrogate for population density. The higher the concentration of these peptide pheromones indicates a higher or heightened concentration of the bacteria that are responsible for them. So even though they're functioning analogously to homoserine lactone, functionally they're doing it in a very different manner. Remember that homoserine lactones were freely diffusible across the membrane going out and freely diffusible inside. These peptide pheromones never come back into the cell. Their fate is to remain completely outside of the cell. What the bacteria do in order to sense those is they use what's referred to as a 2-component system. We're going to talk extensively about 2-component systems in another part of this section, but for the moment just realize that a 2-component system exists of a histidine kinase in the membrane and a response regulator existing as a transcriptional factor in the cytoplasm. We've talked, in essence, about another form of 2-component system when we talked about chemotaxis. So as you're studying about chemotaxis, remember the key A protein is a histidine kinase. That histidine kinase is analogous to this histidine kinase. It's just that in chemotaxis, that histidine kinase is entirely inside the cell. It communicates with the outside via that methyl-accepting chemotaxis protein that detects chemoattractants or chemorepellents. In the case of most histidine kinases, so key A is actually a very unusual histidine kinase, and key Y and key B are very unusual response regulators. When we talk about 2-component systems and histidine kinases and response regulators, for the most part, we're talking about proteins that function just like the two that I'm going to describe here. So this histidine kinase is a membrane-bound protein. It has a domain that is exposed to the exterior environment, and it has the ability, that exterior domain has the ability to bind to that peptide. Non-covalently, it just recognizes it, lets it go. But the more concentrated this cellular dependent signal is, this peptide pheromone, the more frequently the histidine kinase is engaged by that peptide pheromone. And just like HEA, it will phosphorylate itself on a histidine on the in this case on the cytoplasmic side. So that's not shown very well in this cartoon, but these boxes just to the left of the histidine kinase are showing that a histidine has been phosphorylated, and then that phosphoryl group can be transferred, it's actually stolen, by an aspartic acid on the response regulator. This is very much like key Y becoming phosphorylated or key B becoming phosphorylated. But remember, key Y and key B, their effect was in the phosphorylated form to interact with another protein, either at the basal body in the case of key Y phosphate or the MCP in the case of key B phosphate. Those are very unusual response regulators. Most response regulators, when they become phosphorylated, when they become activated, will behave exactly as this response regulator does here for quorum sensing. This phosphorylated response regulator now has the ability to bind to the DNA, to specific sites associated with promoters for quorum-dependent proteins. So in exactly the same way, this response regulator is going to bind. It's going to produce more of the histidine kinase, the peptide pheromone, and the response regulator. So it allows, again, the system to really ramp up very rapidly to make sure the entire population gets the message in a hurry. But it also increases the transcription and then the translation of proteins like toxins or enzymes that need to be secreted. So in essence, this is exactly the same thing as going on in gram-negatives, but you can see that virtually there is no similarity in the mechanisms between these two forms of chemotaxis. Right, so the quorum is detected as peptides accumulate, not homoserine lactone. This cartoon is just a little bit better. I found this more recently. But again, it shows that the peptide signal is being produced in the cytoplasm, and it's chopped up into pieces and transported into the exterior environment. So here, at low cell concentration and low signal peptide concentration, the kinase is not being phosphorylated very frequently. So the response regulator remains unphosphorylated, and so you have virtually no expression of the quorum-dependent proteins, shown here as the red box with no expression. Now, over time, as the population increases, you now have an increased extracellular concentration of that signal peptide because all of the members of the population are making that signal. So now the kinase is much more likely to be engaged by a peptide, which results in a phosphorylation cascade where the phosphoryl group is taken by the response regulator, which now becomes active, and now you get the expression of the quorum-dependent proteins. Now, to give you a more concrete example, especially one that's important in terms of pathology, in terms of how bacteria cause disease, this is a system. This is one of this is perhaps the master regulator in staphylococci of the ability to produce toxins. So staphylococci cannot be if you knock out or mutate this pathway. This pathway is so important to its virulence that it will not function as a pathogen without this. And it illustrates all of the different examples that we've just talked about. So the autoinducer peptide, so that's what AIP stands for here. These are little, they look like little sperm cells or something, but in reality, these are cyclic peptides. So these are little breakdown products of a protein here in the cytoplasm. This AGRD protein is a peptide or a protein that will be chewed up and secreted by AGRB, releasing these autoinducer peptides. Once the autoinducer peptides reach a very high level, indicating that there are a lot of staphylococci in the environment, they'll interact with this histidine kinase. This specific histidine kinase is called AGRC. You can see that it has several domains that interact or that are exposed to the exterior environment. So one of these domains would interact with the autoinducer peptides, which would phosphorylate a cytoplasmic histidine, a highly conserved histidine. This is found in roughly the same position in virtually every histidine kinase ever described. It becomes phosphorylated. There is a transfer of the phosphoryl group to the response regulator, AGR-A, which now becomes active and has the ability to stimulate two promoters. So promoter 1 is not shown here. Promoter 1 is a constitutive promoter, which is producing, even at low cell concentration, is transcribing AGR-B, D, C, and A. Recognize that these genes are those genes that are responsible for producing the autoinducer peptide, the molecule that transports them across, as well as the two-component system, AGR-C and AGR-A. Those are all controlled. So those are going to be expressed by P1, which is not shown here, a constitutive promoter which expresses that operon at very low levels all the time. But now, when AGR-A becomes activated, which only occurs when there are a large number of bacteria in the environment producing these autoinducer peptides, it turns on both promoter 2 and promoter 3. You can tell from the direction of the arrow by turning on promoter 2, this is going to ramp up the quorum sensing system. It's going to speed this system up in the entire population by increasing the amount of all four of these proteins, including the two-component system, as well as the transport system and the autoinducer peptide. So it's going to ensure that the entire population gets the message very soon that quorum has been reached. But in terms of virulence, virtually all of the virulence determinants of Staphylococcus are controlled by a regulatory RNA, RNA3, shown here. So that's the gene that is transcribed in the opposite direction from promoter 3. Now, RNA3 is going to be expressed, for the most part, only at high cell density. And RNA3 now has the ability to go throughout the genome and control messenger RNA translation. And so when quorum is reached, the bacterium will now begin to produce exotoxins that it wasn't producing when cell numbers were low. And it will also begin to decrease the production of surface proteins. So the bacterium will go from a behavior where it's attaching to something like cartilage or bone or something, in an infected site. It will go from that type of a physiology to one where suddenly it stops producing those, so it can change its behavior in terms of spreading in a host, but it's also going to produce exotoxins as well. So this kind of quorum sensing is a master control for virulence in Staphylococci. This is just another one, picture kind of showing the same thing from a company that's proposing a way to interfere with quorum sensing as a means to potentially interfere with infectious diseases by Staphylococcus aureus. So here, those autoinducer peptides, and this is not to scale, these peptides are very small. The autoinducer peptides are shown as these red little sideways P's here, and staph are shown as the golden circles. So at low bacterial density, quorum sensing is off. When that happens, as I said in the previous slide, the adhesion genes are expressed. So without that RNA3, the genes that allow the protein to bind to fibronectin and parts of the extracellular matrix of connective tissue, those are expressed. So the bacterium will express this colonizing phenotype. So it will grow just attached, but it will remain pretty much in that area. Now, once they've reached a certain density and the autoinducer peptide is there, now suddenly quorum turns on the expression of virulence. So now they begin to produce toxins, things like homolysins. You've seen evidence in the laboratory of Staphylococcus homolysin already. If you've looked at the Staphylococcus aureus plates that we grow, the blood agar plates, if you hold those up to the light, you can see the red blood cells are being released. What's happened is by quorum sensing, those colonies have produced enough of this autoinducer peptide that they have now begun the production of homolysins, a secreted protein toxin that's lysing red blood cells. In reality, the staph doesn't care about lysing red blood cells. It's trying to lyse white blood cells, neutrophils, lymphocytes, things that are designed to kill it. But it will also, as an innocent bystander, lyse red blood cells. Hence, we call it hemolysis. but they release lipases and things like that. This allows the staph to become invasive. So it leaves this kind of quiet quiescence where it's just growing on the surface of connective tissue to now actually be able to invade and to spread. And this is when the disease becomes particularly virulent for the host. This particular company that I stole this slide from, and I don't credit them, and I should have perhaps, I don't remember who they are, came up with a potential way using an antibody that they had made against this autoinducer peptide, with the idea being that they could block staphylococci potentially from changing into this invasive phenotype. In theory, at least, this is a really good program. I don't know whether this has ever been shown in principle to work, at least not in humans. But the slide is nice because it kind of shows how this system works. Okay, so in the last minutes, I've got 10 minutes left, I want to talk a little bit about the original bacterium that Ken Nielsen began to describe that really led us to the discovery of quorum sensing. It's a bacterium that lives naturally as a non-pathogenic organism in marine environments around the world, Vibrio fischeri. It's related to the bacterium that causes cholera, so it's related to a human pathogen. But for the most part, it exists as a mutualist with many marine species, both vertebrates and invertebrates. It's been studied mostly in squid, so we know it mostly as a mutualistic bacterium, so helpful to the squid, and the squid are helpful to it. I'll show you, or actually I'll actually have someone else narrate and show this. Members of the genus Eupremna, which are little cute squid that live in tropical barrier reefs. And this association is non-pathogenic. It's actually beneficial to the squid. So this is a video. Oh, not yet. I forgot what slide was next. This is the mechanism whereby that underpins this mutualism. So I mentioned earlier that the quorum-dependent proteins are frequently secreted. This is an example of quorum-dependent proteins that are not secreted. But what's happening here is that you have Luxi, and this was the first bacterium that this was discovered in, and it was named Luxi, produces a homoserine lactone. This is one specific for Vibrio fischeri, so this simply describes this 3-oxo carbon 6 homoserine lactone. Just means that that's the specific substituents of this molecule that diffuses out. It reaches a high concentration, it diffuses back into the cell, it binds to the repressor protein LuxR and turns it into an active dimer, which now has the ability to bind to this Lux box, this DNA, which simultaneously shuts down the production of LuxR. You can see that the transcription, this dashed line is meant to indicate that transcription is decreased of the repressor. But it's actually simultaneously activating the production of LuxI, the synthase, which makes even more of the homoserine lactone, so the entire population gets the message. But at the same time, it's increasing the transcription of this operon, Lux C, D, A, B, and E. I don't know what all of these genes do, but A and B, these two genes together, form a luciferase, very similar to firefly luciferase. So this bacterium will now produce light. But it doesn't make sense for one bacterium to produce luciferase because it's almost like a Zen question. You know, what's the sound of one hand clapping? How much light can one little tiny bacterium produce? Not enough that you can detect, and not enough to be helpful for the squid. So the bacteria have evolved to use quorum sensing to make sure that they only express light when the bacteria are at very high concentrations. This is the video. And I'm sorry I muted
this. They really are beautiful animals, really smart animals. They're largely the body size.
It's hard to hide in the ocean, but the Hawaiian bobtail squid is a master of disguise.
During the day, if they just get into the sand, they'll come out and just sit on the surface with a sand coat on them, trying to be invisible. And if that doesn't work, they'll eject out and leave that blob of ink behind in their shape.
Their camouflage strategy at night is even more amazing. Luminous bacteria actually live inside the squid's body and light up its underside, helping it hide from predators.
So there are many ventral light. They're camouflaged against the moon or the starlight so that they don't cast a shadow. And so it's not casting a shadow, it's like a cutout protein device.
support from the National Science Foundation, biologist Margaret McFall Nye studies the squid's remarkable light organ, small pouch that's home to the cloaking bacteria Vibrio fischeri. Its structure has a lot of similarities to an eye.
We can look at all parts of the light organ, so all parts of where the bacteria go into the light organ and then reside.
To better understand how the bacteria take up residence inside the squid, McFall Nye's team studies the babies under the microscope.
These protrusions right here, which we call appendages, help to filter, basically bring in bacteria from the seawater. So, you kind of have this little cave, this kind of nest that the squid makes for the bacteria.
We bring the eggs into the egg room.
Nelba Karras goes to Hawaii 3 or 4 times a year to collect adult squid off Oahu. Back in the lab, she oversees the squid conduct and the nursery.
I make the squid every 2 weeks. A female will generally lay eggs about every 3 to 5 days. This clutch is a fairly good size, probably 200 eggs or so.
Analyzing this squid bacteria symbiosis could help develop new camouflage materials for the military, or lead to new pharmaceuticals to fight bacterial infections.
At the very basic level, we're asking how animal cells talk to bacterial cells.
A conversation worth listening in on. For Science Nation, I'm Miles O'Brien.
So they didn't really go in. So this is a really cool system. So Margaret, who has since moved from Wisconsin back to Hawaii very smartly, this process whereby they use mutualistic bacteria to produce light, not to cast a shadow, is known as counterillumination, which is a really cool system. So the way this works is that the squid is nocturnal. During the day, because it's just this small little bite-sized piece of calamari, it hides in the sand to keep from being preyed upon. And then it comes up at night. What's going on during the daytime is that light organ now is serving as a culture tube, a flask for Vibrio fischeri. The Vibrio fischeri are growing over the course of the day. And the timing has evolved such that now at exactly nightfall, as the squid rises up out of the sand, the bacterium have reached a population where they're at quorum. And so now the entire bacterial population, when it was lying in the sand, was not emitting light. But now, once it reaches quorum, which is at the same time that the squid begins to hunt because the sun is down, it's emitting light. So now this squid can go up into the water and begin to hunt. And this counter-illumination strategy is really clever because, predators, eels and such lying at depth, one of the ways that they detect, a night's tasty morsel above them is on a moonlit night or a starlit night, something casts a shadow. So all animals have the ability, really, I think, to detect shadows. If you've ever noticed, I had a friend who raised quail, and the quail would suddenly in their yard, if a bird flew over, if it cast a shadow, they would just huddle to the ground. They just reacted to the shadows. So predators have evolved to detect a shadow as something swimming above could be eaten. Instead, just carry a moon with you. So these squid have got the ability to actually modulate how much light that their bacteria are emitting. And they can match it to the moon, to the phase of the moon. So during a full moon, their light organ produces even more light. So anyway, these squid hunt for the night. Sun starts to come up in the morning, they go home, they go to bed, they carry themselves in the sand, and they have musculature around that light organ that they can squeeze that light organ. And they squeeze almost all, like 95% of the bacteria out of that light organ with all of the waste products as well. The remaining 5% will grow over the course of that day. Again, reaching quorum just in time to match the hunting cycle of these squid. So it's a really beautiful, highly evolved system, this counter-illumination system. This is I'm probably not going to take the time to go into this, but this is just the developmental biology and how these bacteria get in, because the bacteria are not, this is horizontal transmission of symbionts. They're not getting these bacteria from mom or from dad. They're actually acquiring them in the environment. And so they're swimming in through these pores as these squid are still developing. And the squid has actually evolved the ability to make sure that the only bacteria that can get in there and survive are Vibrio fischeri. So they're these squid, as cute and adorable as they are also probably the best microbiologists I've ever known. They can maintain largely a pure culture almost exclusively. Here, one last video, and then it's the weekend. And this is Sir David Attenborough, so this is worth listening to.
Turn off the submersible headlights, and you see a pyrotechnics outside. These lights are created by animals. This is bioluminescence. A deep sea anglerfish flashes in the dark. The light is generated by bacteria that live permanently inside the lure, which attracts prey to these murderous teeth. There are all sorts of lures out in the darkness. And what is the purpose of this lure, suspended on the long rod, one of the turning to find Sept teeth? It's difficult to be sure. But then this monster does have another giant flashing lure much closer to its mouth. These fish are called anglers because they use their lures in much the same way as fly fishermen use their imitation flies. For a hunting squid with huge eyes, this lure is intriguing. It might just be food.
It doesn't end well for the squid.
A satisfying meal for fish with a highly extended stomach.
So same bacterium can be mutualistic with vertebrates as well. Have a great weekend. I'll see you guys on Monday.